This video is going to answer 3 questions at the same time. The first question in Chapter 11, Review, explain how gametes are produced by meiosis. The second, explain the purpose of meiosis. And 3, show crossing over on the chromosomes shown below. How does crossing over lead to genetic variation and evolution of a species? In essence, question 3 is also asking you about question 16. So the first question, how gametes are produced. The first question is about meiosis. Look, I got a better pen. And with meiosis, it's important to remember that 2n equals 46. The end result of meiosis is that we're going to end up with cells where n equals 23. This is human meiosis. If it was a different animal, our diploid number, the 2n number, diploid, and our haploid number, haploid, those would be different if it was a dog or a cat or a fruit fly or a plant, but it all works out the same way. So with meiosis, we start with one cell, which we're going to say is diploid. We just said that a minute ago, diploid. And I'm going to draw in a couple of chromosomes just to represent the chromosomes here. So you'll remember this drawing from class. We'll fill in the chromosomes in just a minute. But from class, we said that the cell will go through interphase, and during interphase, something really important happens. Hopefully you'll remember this. S happens, the S phase. And during the S phase, DNA copies itself. So if you look at the cell at the top, we have a new chromosome. That would be its sister chromatid for that one. And then this red one is also going to copy itself. Now, prophase, metaphase, anaphase, and telophase. We go through a second round of division, but in this time, there's no interphase. And if there's no interphase, there's no S phase, which I'll just abbreviate with NS. No S. Okay? Now if there's no S phase, well, what's happened to these chromosomes up there? They didn't copy themselves again, but they did do something magical. And we need to look at that right here in this little arrow. We'll draw it nice and big so you see what's happening in prophase and metaphase. These chromosomes are lining up. I'm going to draw them like the X's now that they really are. They're lining up side by side. And these are what we call homologous chromosomes. And those homologous chromosomes form, lots of new words here for you, a tetrad. All of these words are in the vocab list in your study guide. The thing that happens, and it's going to be very difficult to draw, but this is question 3, but the thing that happens at this point in time is called crossing over. And I'm going to save crossing over for a second slide. But in short, what it means is that the white chromosomes and the red chromosomes sitting next to each other, they're kind of smushed all together, and then when they're done, and they're going to end up getting pulled apart during meiosis 1, parts of the red one and parts of the white one have exchanged sections. So you've got some red and white alleles on opposite chromosomes. I'll give you a better picture on the following slide. Back to meiosis. So prophase occurs, metaphase occurs, anaphase occurs, and we end up with our chromosomes now. You're going to see I'm going to draw them a little bit different. We've got these chromosomes now that have a little bit of both worlds because of that crossing over. And let's see there. So we see that they've swapped different pieces of their chromosomes, and therefore the alleles and the genes that are on there are different. this does lead to genetic variation, which is another question that's being asked. So what happens now? Well, if there's no interphase, there's no copying of these new chromosomes. We just go through prophase, metaphase, anaphase, and telophase. And now metaphase looks similar that it did in mitosis, where the chromosomes just line up along the middle. Let me make one of these pink and the other one white. Kind of put in our pink and our white bands on there so you can see them. And they line up in the middle, they get pulled apart, and ultimately, whatever chromosome you see there, well, that's going to end up in that one. I forgot to give it a little red stripe there. So it ends up in there. There's its red stripe. The other one ends up in there, and it gets a white stripe that you see. And the same thing is true on the other side. And we end up with really similar but different patterns. Well, you'll notice that these down here, these are all haploid. Why are they haploid? If you count the chromosomes that are in there, you'll notice there's half as many. So there's only one here, right? And one in each there. And each one has only one. And when we started at the beginning, we had two. This would be the same thing if we were talking about 23 chromosomes and 46 chromosomes. In the previous slide, we mentioned crossing over. Crossing over increases genetic variability, and you're going to see why in 1 second. For this, I'm going to use a picture because my drawing is a little sketchy. No pun intended. Now, take a look at this picture. Here are our chromosomes that we were mentioning during prophase and metaphase 1. And they're sitting side by side. You'll see we've got our homologous pairs. You learned this in question 1. Each one of those would be your sister chromatids. Okay? And then something during prophase and metaphase, you notice that they exchange pieces in a variety of places, not just one or two, but it's all over the length of the chromosome. Where during prophase and metaphase 1, they actually wrap around each other. This is called a synapsis. And when they pull apart during anaphase, what do you notice happened to the chromosomes? They have actually swapped pairs. Again, this is really important. And the reason why it's really important is because it leads to genetic variability. I'm going to only spell variation. Variation. I ran out of room. In other words, we have many more opportunities for gene shuffling to occur, and it's this gene shuffling which ultimately will lead us into chapter 16 questions. And gene shuffling is one of the processes of evolution, or how species change.